guess at this point I get to introduce myself for the next talk. My name is Dave Zawieja. I'm a Regents Professor at Texas A&M University, Health Science Center College of Medicine and Medical Physiology Department, and I'll be talking about inflammatory and immune mechanisms in lymphatic vascular disease. Okay. It's going to be my pleasure today to have a little bit of time to talk about some of the underlying basic pathological mechanisms that you'll see will underlie lots of the clinical pathologies later this afternoon. And in specific, I hope to give you an idea of our growing understanding of the relationship between the lymphatic vascular system and the immune system that it serves. And in fact, as of late, we've been in my laboratory, we've been reterming it the lymphatic system as part of the classical immunovascular system, because one of its main causes is to bring things from the tissues to the node and then back from that node to the blood to be distributed out the body. And as we talked about yesterday, and Stan already alluded to, the lymphatic system has critical roles in fluid and macromolecular homeostasis, edema resolution, lipid uptake, and most importantly, inflammation and immune cell antigen trafficking. While I got into this field related to the earlier parts, looking at macromolecular and fluid homeostasis and edema resolution, what I've learned, and painfully in some cases, in the last 10 years, is it's very difficult to dissociate that from the latter parts. That is the role in inflammation and tightly tied to immune cell and antigen trafficking and cytokine trafficking. The bottom line is all of these processes require the lymph to move from one place to another, from the interstitium to the nodes, and then from the node back to the blood. And it's in doing so, the organization of how you do that is what we described yesterday. And what I will tell you is that the processes by which we discussed yesterday, where I alluded to the primary controllers by kind of physical factors of the pressures and shears within the lymphatic system, impacted by neural agents and humoral agents, it's that latter part that we're learning more and more about, even though it's where I first started in the field 30 years ago. And in fact, the latter part of immune cells that produce inflammatory cytokines that then can act on lymphatics to both make them grow more quickly, i.e. developing inflammatory lymphangiogenesis, as well as to modulate the lymphatic function in the larger vessels, like as I discussed yesterday. The bottom line is that you have to take that lymph and move it to the nodes, and then it has to traffic through the nodes. In mechanisms you would think we have a pretty good idea about how they happen, when in fact the reality is we know very, very little about the plumbing, the details of the plumbing of the interactions of the lymphatic system, the high endothelial venular system, and the T and B cell zones within the nodes. And lastly, one of the things that's critically important, and Dr. Roxon showed this in his anatomical preparations, is we have to dispel this idea that lots of people have that lymph goes into one node and the magical immune things happen there and lymph comes out on the efferent side and never sees another node. That's truly true, almost unheard of in humans. You can see that in some smaller animals in very few nodes, but the majority of lymph passes through 3, 4, 5 nodes along its path from the interstitium back to the blood. And so the lymphatics play a critical role as the immunovascular system. They deliver antigens, cytokines, and immune cells to and from the lymph nodes, that is, to the lymph nodes from the tissues, from the lymph nodes back to the blood. And they help to develop not only fluid homeostasis, but also inflammatory resolution, development under the wrong conditions, and immunological homeostasis as well. And when the lymphatic transport system fails, and we think about this classically in the context of what most people in this meeting are thinking about, and what I was classically trained as, that when the lymphatic system fails, it leads to accumulation of fluids and macromolecules. But it also leads to the accumulation of lots and lots of antigens, whether they're self-antigens or foreign antigens, and more importantly, immune cells and whatever cytokines they may normally produce or abnormally produce in those tissues. So that lymph flow is not just a matter of pooling up fluids, but it's a matter of pooling all the things it carries under normal and abnormal conditions. And this is a common cause and part of the resolution process as well in inflammation. Prolonged, and this is an important point that I hope we can, if you get one thing that from this lecture that we get across today, is that prolonged lymphatic impairment and impaired lymph flow, it leads to not only lymphedema from the perspective of leaving fluid behind, but you also leave behind all of those inflammatory cytokines, or the immune cells that have to normally traffic out of those tissues to the nodes, either under normal tolerogenic mechanisms or under activation when you have some inflammatory process, whether it's minor or major going on. And leaving those cells and their excretions behind makes the inflammation much, much worse, because it typically leads to increased permeability of the blood vessels and enhanced inflammation. So what are some of the classic roles of lymphatics in immunity? These are the ones that you can see variably in different immunity textbooks. Well, one that we know of very well is that the lymphatics are the key mechanism by which you transport antigen from the interstitium to the node. Why is that so? Well, first of all, they're the principal transport route for anything larger than a couple thousand molecular weight that is released within the tissue spaces. In order for it to get to the node or back to the blood, it doesn't go into the venules or capillaries. So very, very little of it will under normal conditions. The vast majority of it will be picked up through the lymphatics because of the unique characteristics of the lymphatic capillaries that Dr. Roxon talked about. You have large endothelial gaps that not only let large macromolecules, i.e. antigens, get in from the interstitial fluid, they also allow cells, form particulate matter, to enter into the lymph at the lymphatic capillary. And so they become the predominant means by which you capture antigens from different places in the tissue. Second classic role of lymphatics in immunity is the trafficking of immune cells. We'll skip to the end one. Lymphocytes are named lymphocytes in part because they traffic via lymphatics from the nodes back to the blood as their primary egress route. And so it's very obvious what role and interactions lymphatic play in them, even though we don't characteristically know all the detailed mechanisms that regulate this process. But I would like to focus a bit of today's lecture on, and that, in fact, it's another important whole cadre of immune cells, that is the innate immune cells, or those that transition from innate immunity to adaptive immunity, that are also trafficked in the lymphatics from the interstitium back to the nodes. So these are cells like professional antigen-presenting cells, like dendritic cells, Langerhans cells, macrophages, and of course some of the lymphocytes as well. And I'll just give you a brief overview of some of the lymphatic characterization from the perspective of the lymphatic plumbing of the node. So in every node, and this is a classical kind of node, though they don't all look like this, of course, you will have multiple afferent lymphatics entering the subcapsular sinus, and then you will have usually one or just a couple of efferent lymphatics that lead the medullary sinus on the node. In between them, of course, you have all the T cell zones and the B cell follicles and lots of medullary and intermedullary sinuses that somehow connect the subcapsular sinus where afferent lymph is coming in to the medullary sinus where lymph is going out. In addition, and importantly, you have the blood supply that goes to the node, and particularly the high endothelial venules, the unique blood supply that brings the naive lymphocytes into the lymph node in order for them to egress there and then be trained to react to the appropriate immune responses. This is shown here characteristically in a kind of a cartoon diagram, and one of the things you notice in the previous one and this one, and if you look in 10 different immunity textbooks, you'll find different versions where you see this nice afferent lymphatic coming in here, you have a nice efferent lymphatics going out connected to the medullary sinus, and you have this beautiful subcapsular sinus that somehow connects directly to the medullary sinus. The reality is we don't know or understand what all these plumbing connections are yet. And they're very, very important because under normal non-immune response conditions, you need to allow immune flow to go through this node and allow some mechanism for the lymph node to sample typical self-antigens and produce a self-tolerizing response, but let large lymph flows go through the node without impeding it. Then under appropriate inflammatory reactions or responses with a foreign antigen out in the tissue, you have signals coming in here that will, in essence, allow some of the lymph flow to go through, but what you see is the node swells. The node swells quickly, and then it swells even more over a prolonged period of time. That initial swelling within a few hours to the first day or two is not what most immunologists think, that it's T and B cell proliferation or accumulation. It's actually swelling of the fluid structures of the lymphatic node, lymph node as well. We don't understand that processing exactly how it works, but we know that in fact this is the early case. Later on, as that immune reaction occurs, then the T and B cell zones proliferate and accumulate lots and lots of cells, and you get further dramatic increase in size of the lymph node, but in that case it's cellular sizes. And so that very important difference between high lymph flow through this node with a little bit of information going through the B and T cell zones to provide a tolerizing signal versus under normal conditions versus kind of retained structures where you get more lymph flow of antigens into the T and B cell zones to allow the appropriate training of the T and B cells for the mounted adaptive immune response. Those mechanisms of how that occurs and importantly how we switch, remain to be seen yet. So let's talk a little bit about what is the population of the cells that we typically see in lymph. We have a very good idea, because it's been studied for many, many decades now, what the immune cell population in postnodal lymph is. And it'll vary slightly in percentage-wise from animal to animal, but generally speaking, what you have is 75% or so of the immune cells in postnodal lymph are lymphocytes. You have a small but significant population of the innate immune cells, that is neutrophils and macrophages, with a very low population of dendritic cells within postnodal lymph. So then the question is, what do we have in prenodal lymph? Well, if you look in the literature, there are a lot of papers that prescribe to say they've captured prenodal lymph or some analog of it and measured the immune cell population of what those are in there. The problem is, as you see in the next couple of slides, what is prenodal lymph to one large node, which is typically where they're collecting from, because they go to a large node because it has large lymphatics, easier to cannulate and easier to collect lymph from. But the reality is that afferent lymph to that node has already gone through one or two nodes beforehand, smaller nodes upstream that aren't seen within that specific preparation. And so what they're really sampling is not true virginal prenodal lymph, but it's lymph that's already gone through a couple nodes. So the reality is we're not exactly sure yet what good prenodal lymph population of immune cells are. We know from some work that has, where it's predominantly prenodal at least, that there's a much higher population of the innate cells, particularly the antigen-presenting cells like dendritic cells, Langerhans, and macrophages. But they still do have they appear to have a pretty significant population of lymphocytes. And that concept of the fact that lymph is not just simply going through one node and then goes right to the thoracic duct to be distributed back to the blood is kind of schematically diagrammed here. And we won't spend a lot of time on it, but here's the focus on the one on the left, and what you see is that at every node you can have multiple afferent lymphatics, some of which may be true prenodal, never seen a node yet before. They get processed and you get an efferent lymphatic, but you can have another afferent lymphatic that has not gone through the node, enter that afferent lymphatic or a new one, and then eventually you can see that all of these will go through multiple nodes on its route to blood. Presumably this is a fashion in which it's been designed to enhance the ability to uptake self and foreign antigens and allow the generation of the appropriate immune response. Let's focus on some of the cells that are in the innate immune path, because these are the ones that are less well described historically, but which play a critical role in the development of a typical immune response. And so one that we know a fair amount of is on the transport of dendritic cells. As Dr. Oxen alluded to earlier, dendritic cells and antigens will enter the lymphatic capillaries through the primary lymphatic valves, or these holes depicted in this lymphatic capillary structure here, that are located between these point button contacts of junctional complexes in the initial lymphatic endothelium. Those button holes and those rearrangements of that endothelial junction proteins, there's some data that's suggestive that the dendritic cells actually rearrange it in that fashion for them to enter based on certain chemokine gradients, usually CCL19, CCL21, secreted by lymphatic endothelium. When those DCs come to a junction, they can rearrange it and essentially make those gaps, make those primary valves or those portals. And from there on in, if you have enough immune cell flux, those become the predominant portals, or in essence, the primary valves of those lymphatic capillaries. Once they get into the lymphatic capillary, in this case, they're all, of course, crawling. And as they get into the initial lymphatic capillary plexus, they're still crawling because lymph flow and the lymph velocities that Dr. Roxon had in that really tiny text in that big chart are really, really low. They're about 2 orders of magnitude in terms of velocities from the flows I described the other day, which are about an order of velocity lower than typical blood flow in a small arterial or venule. So it's very low flow, so they can't hitch a ride on flow at this point. They're literally crawling along, and they crawl along because there's enough flow to provide a chemotactic gradient that they can follow to get into larger and larger lymphatics, particularly the precollectors. Once they enter the point of getting into the collecting lymphatics, where now you've got a significant flow generation, significant shear, they can release from the endothelium and in essence carried along by lymph, and within a matter of a couple of minutes at most, they'll be delivered to the next downstream lymph node by the contractile lymph pumping. And this is described here a bit. I won't go into great detail since it's a matter of time, but if you just look at this series of images here, what it's showing is the opening, this is staining for lymphatic endothelium. You can see this opening in here growing basically as one of these dendritic cells crawls through this interendothelial junctions in the lymphatics, expands it, makes it bigger, and then crawls through to get into the lymphatic lumen itself. So let's talk about another critical thing that I've already brought up, is that antigens are transported preferentially through the lymphatics from the interstitium to the node. We talked about this in comparison of their size. Once you get into the subcortical space delivered there by lymph nodes, then these antigens will be distributed through the various conduits and parts of the lymphatic plumbing that will allow the appropriate cells, the antigen-presenting cells, particularly the professional antigen-presenting cells there, that will sample that, and if they're primed for it, will then develop the appropriate T and B cell response. In particular, molecules that are smaller than 80 to 100,000 kilodaltons will percolate through that subcapsular sinus into the conduit system that are then in direct contact with the dendritic cells and the network of T cell zoned areas. And in fact, we now know they're in contact with the B cell zones as well. What does this mean? Well, that's a great way for delivering foreign antigens to the cells of the naive cells of the node. It's also a great way for delivering foreign antigens to cells that are learned to adapt to a particular foreign antigen response. And that's a classical idea that lymph-carrying antigen is thought to behave. But recently, in the last 5 to 8 years, there's been this growing idea, and it's not a new idea actually, that lymph may not provide only the foreign antigen information path, that lymph actually is the best body fluid to carry self-antigen information to the nodes. And it delivers that self-antigen information in a process to the nodes to say, with the appropriate cytokines that we do not yet understand, that this is me, do not mount an immune response against me. Why is that? Well, as Dr. Roxon alluded to yesterday, that lymph is not just simply an ultrafiltrate of serum. It starts that way, but then it's modified in every individual tissue by the surrounding parenchymal cells in that tissue. And they release their breakdown products as those cells live and die, apoptose, and release parts of their self out into the interstitial fluid. Those are further broken down by fibroblasts and other immune cells within that parenchyma. And those breakdown products are carried in lymph into the initial lymphatics. And what people don't normally recognize is when you put a foreign tracer or a tracer antigen in your toe, you're going to see it in your popliteal node within a couple of minutes. So that antigen trafficking is very, very fast. Whereas the cells, that antigen-presenting cells in your toe that picked up that antigen, they will take many hours to get from there to the lymph node because they have to crawl along a long distance before they can catch a ride on lymph. And so there's been a number of proteomic analyses recently, in particular one by Dr. Sanambrogio, a series of them done by Dr. Sanambrogio, that took this old idea that lymph is perhaps the best self-tolerizing panel of proteins to give to the naive node to teach yourself not to mount an immune response to it, and has done some beautiful work to document that further. Unfortunately, we don't know what the key mechanisms are that switch between that normal process and the non-normal process when you then have to mount an immune response to a foreign antigen mechanism. So I'm gonna, for the sake of time, I'm gonna skip over this a little bit already, once some of it, and that is as you bring in lymph, you can bring in antigens as well as antigen-presenting cells carrying that antigen. These antigens get there quickly in a matter of a few minutes to tens of minutes. These APCs that captured the antigen in the interstitium had to crawl all over the place and through the initial lymphatic plexus instead of being carried. They will get there as well, but they'll take hours to get there. When those antigens, whether they're self or foreign, will come into the subcapsular space, they get interacting with the professional antigen-presenting dendritic cells that lie here and then interact with the T and B cell zones to mount the typical response. And that's shown as well here in a schematic way. Again, this plumbing idea of where lymph flow goes is rather simplistic, and in fact, we don't believe it works this way. It's a lot more complicated than that. What we do know is that the T cells, they'll wander back and forth looking for the appropriate, the B cells and T cells will wander around looking for the appropriate responses, and then they'll get the right pathways, and those involve SIP, to get into the intermedullary cortical sinusoids, which eventually do connect with the medullary sinuses, and then they become the lymphocytes that egress out of the lymph, trained to attack particular antigens, and then they get delivered into the blood and distributed about the body, where they get home to the right tissues and egress and mount immune response in that particular tissue. I'm going to digress here a little bit. So these are all the classical mechanisms, some of them pretty new, some of them rather old, that people have described in terms of the roles the lymphatics play in the initial inflammatory response and then in the later immune responses. And I'm going to show some data that we recently got in the last few years that shows that there's actually some other mechanisms going on that have not been described. And that is that in addition to the fact that there are these dendritic cells located in the tissues that capture antigen, crawl to lymphatic capillaries, enter, travel via different mechanisms to get to the node, and antigen that's carried quicker from the tissue that gets delivered to that node. Those antigens actually along that pathway get exposed to all the lymphatic surface area within that lymphatic vascular system, which is actually quite substantial. And they get exposed to that lymphatic surface area, the LECs and whatever else is there, very quickly, much quicker than the antigen-presenting cells that captured it in the tissue and have to crawl there. And so could the prenodal, we had an hypothesis based on some preliminary data, could these prenodal lymphatics actually play some earlier pathway in mounting an immune response? The bottom line is that don't pay much attention to it except for what's in yellow. When we compared small mesenteric lymphatics with small mesenteric arterioles and small mesenteric venules of rodents and large lymphatic vessels to large veins to large arterioles and did a genomic analyses or transcriptomic analyses, the bottom line is we were looking for certain things related to the lymphatic endothelium and muscle that would differ between those arterioles, venules, and lymphatics. And we found some of that, but the reality is what we found from the gene ontology mapping, which tells you what the most significant differences are all related to immune response. This is where I began to change my mind about the lymphatics as purely part of the cardiovascular system maintaining fluid homeostasis, that perhaps in doing so, they actually maintain important immune function because the differences that were predominantly found are related to antigen presentation, antigen processing, immunoglobulin signaling, and immune response to infections, et cetera, et cetera. So we began looking for, in these real, true prenodal lymphatics, these are lymphatics that carry lymph that has never seen a node. This is in rat. And what we identified from using that transcriptomic data is that this is a lymphatic vessel. It's a rat mesenteric vessel. It's approximately 100 microns or so in diameter, so it's very small. But it's muscularized, and it contracts and relaxes, and it's just like the one Dr. Roxon showed earlier. And it propels lymph and has relatively high shears. It's located, maybe in this case, about 2 centimeters from the mesenteric lymph node. It hasn't seen any lymphoid tissue. The lymph in this vessel hasn't seen lymphoid tissue before this point, except what exists in the Peyer's patches in the gut wall. And what we then did is stain for one of these transcriptomic, highly upregulated mRNAs that codes for this particular MHC2+. And so it's an MHC2+ is one of the key factors related in professional antigen presentation, and what you see is there's this enormous population of cells that are in this lymphatic. And when I say in the lymphatic, I don't mean in the lumen, because these vessels have been flushed. They're isolated from tissues. And what these are cells. These are dendritic / macrophages. We're still fighting with the immunologists on exactly what to call them. But they're antigen-presenting cells that lie in the wall of the vessel. You can see them here. Where we've stained, in this case, the muscle cells are stained in red for alpha vascular actin. The MHC2 cells are stained in the wall in green. And in this case, the MHC2 walls are stained in the wall in green, and the lymphatic endothelium is stained in blue. And what we know from doing analyses is, and I'll skip over this, is that these cell bodies lie mostly above the endothelial layer, below the muscular layer, but their processes, and that's what's shown here in these 3D representations, actually extend down to the sub-interendothelial junctions. So in theory, they can sample antigen carried in lymph. These have never been described before in muscularized lymphatics. People assume that dendritic cells only came in the capillaries and then existed and did their work in the node. And what we're saying is both of those are true, but along the way, this whole lymphatic tree has a vast network of innate immune cells that are professional antigen-presenting cells that can capture antigen. And I will skip over here and show you that this is an example when we put a foreign antigen in the gut wall, looked at those vessels. So you can see the green is the foreign antigen, this is the edge of the lymphatic, here's the mesentery, and then this is the lymph is carrying that foreign antigen, and you can see one of those cells highly outlined in green because it's picked up that antigen in a matter of a few tens of minutes from when we injected it in the lymph into the gut wall and then carried in lymph. You can see another higher magnification picture of one of these cells within the valve structure of the lymphatics, where this is the actual valve, these red lines. And so they can pick it up and they can process it. And this I'll skip over, but this is to show that they actually process it like antigen-presenting cells. So then what do they do with it? Well, that's the part that we're at now. We don't exactly know, but what we do know is if these cells have been pre-immunized to that antigen, those cells will pick it up and they will egress into the lymph, get to the node in a matter of a few minutes. So they will arrive there hours before the ones that captured this antigen in the tissue spaces, in this case right out of the gut wall. And so we believe that they're involved in prepping the node for what's to come. And we think that they're involved in releasing cytokines into the lymph that are carried to the node that then modulate that lymphatic function in the node to change immune function from a tolerogenic mechanism to a foreign antibody response. I'm going to end with a couple of other slides that show it's not just these particular cells that have a very intimate interaction with the prenodal lymphatics. You don't see these same cells on the postnodal lymphatics typically. In some you do, but to much lower degree, but you see them a lot in prenodal lymphatics. So in addition to APCs, dendritic cells, macrophages, both M1 and M2 types, there are eosinophils, neutrophils, mast cells that are heavily invested along these lymphatics. And all these cells are involved in, we believe, in modulating the innate immune response and transitioning what happens in the downstream node into developing the appropriate adaptive immune response, whether it's tolerogenesis or foreign mechanism. Okay, lastly, I'm going to show you the concept that lymphatics have been classically associated with inflammation. It's actually how I got into the field 30-some years ago. And I've taken a long route to come back to it, but in fact, what we know is that inflammation, lymphatics have been involved with inflammation from the aspect predominantly originally from the fact that they help resolve the edema, or what we learned later is they can help form that edema if their transport is impaired. But we also know now that they may be involved not in just the edema process, but because you trap that fluid, you no longer are carrying the normal immune cells that might be carried by that fluid from the interstitial spaces and what they release, the cytokines within those interstitial spaces, back to the node. And so the lymphatic vascular system is really an immunocentric vascular system that's vital to immune cell antigen trafficking as well as the different parts of inflammation, development and resolution. We've done a bunch of work in IBD. I don't have time to go over it, but I'll just skip to one important thing is that if we look at the development of IBD in this animal model, one of the first things we see within, so this is a IBD model where you inject TNBS, a haptenizing agent, to develop basically a foreign immune response to your inherent proteins from your gut wall. And what you see very early on is that lymph flow falls within the first 24 hours after injecting this haptenizing agent. When lymph flow falls, you start seeing the immune cell density in lymph go up. And this has been alluded to by the original work by Dr. Moskowitz and Krohn long ago when they talked about the lymphatics being one of the key routes to disseminate inflammatory bowel disease. We have gone recently to show that we believe that it's a key route in the installation of inflammation and the development of the changes from acute phase where it can be resolved to a chronic phase where it cannot. Along that early time point, you also start seeing clots form within lymph. These are lymphatics within the mesentery, and you see that they have these clotted tissues in it. So in summary, we think that the in situ lymph falling within this early phase of the IBD development actually helps develop the edema in the gut wall. This helps prevent the movement of the inflammatory cells out of the gut tissue, trapping them there, trapping the cytokines, and developing a long-term inflammation. And if you can't develop the mechanisms to resolve that and remove it with lymph flow, you can transition that from an acute phase to a chronic phase of inflammation in the gut wall. We're developing some similar concepts in terms of what happens in the skin during various skin inflammations, chronic and autoimmune, and it appears that there's a similar process going on there, as well as in UTIs, in urinary tract infections. So, in summary, I'll end up by saying that the role of lymphatics in both inflammation through its classic ideas and through some more recent ideas that I've described today, and the relationship to immunity is a very, very important one. And inflammation has one of its hallmarks, swelling and edema. Lymphatic vascular system is known to play important roles in preventing or resolving that edema, and if you impair it, in actually generating that edema. Lymphatic vascular system is the preferred route of transport of large macromolecules, anything over a couple thousand molecular weight from the interstitium to the node, which includes basically all antigens. The lymphatic system is an immunocentric transport system that's vital to immune cell and antigen trafficking. There's a resident population of immune cells that lie in or adjacent to the wall of the lymphatics that can help, we believe, help modulate lymph flow during inflammation and thus change the pattern of immunity. And that they carry immune cells, classical ones we know about, but they have a lot of the other ones, innate cells, lying right alongside them that appear to play critical roles in changing lymph flow depending on what the cytokines that are being responded to at that point in time can influence then lymphatic function. In closing, a better understanding of these interactions of immune cells on lymphatic function and lymphatic function on immune cells during inflammation and how these interactions and transport change during inflammation is critically needed, because a lot of the patients you see with chronic edema will not only have chronic edema but a low-level inflammation within that tissue that needs to be resolved, and lymph flow is, we believe, one mechanism. Reestablishing normal lymph flow is one mechanism by which that can occur. At that point, thanks.